Welcome to Biology 1408. This is Chapter 7, The Cellular Basis of Inheritance, from the OpenStax textbook, Concepts of Biology. In Chapter 7, we have several objectives we are going to study. Some of those objectives include learning about the variation among offspring and how that creates a potential evolutionary advantage for reproduction in that population. We're going to look at 3 different types of life cycles. We're going to describe how the chromosomes and cells behave during a process called meiosis. And then we're going to look specifically at the mechanism of meiosis and how it creates genetic diversity in its gametes. We're going to try to look at and compare meiosis to a process we call mitosis, which we learned about in Chapter 6. And then we're going to look at what happens if there's a mistake or error that occurs in the process of meiosis and how that leads to certain genetic disorders. In sexual reproduction, the idea behind having variation within the gametes is to produce offspring that have better ability to survive and reproduce within that population. Again, variation is introduced by the process called meiosis, and this gives those species a higher advantage in the reproduction and survival of that population. The 3 types of sexual reproduction life cycles that we're going to go through are called diploid dominant, haploid dominant, and alternation of generations. And these 3 types of life cycles occur in different types of organisms, of animals, fungi, and plants. So let's talk about the diploid dominant life cycle first in animals. The diploid dominant life cycle is called diploid dominant because the organism that we typically see as the organism of that population, in this case for showing humans, is diploid for the most part. There are specialized cells in the gonads of the male and female, that's the testes and the ovary, that are called germ cells. And these germ cells will go through the mechanism called meiosis, which creates 2 types of cells, the egg and the sperm. When the egg and sperm come together and fertilize, they create the zygote, which then can go through mitosis and produce more adult versions of that organism. Therefore, meiosis is very important in the life cycle of most animals. In fungi, their dominant life cycle, or dominant portion of their life cycle, is the haploid, meaning that the majority of the time that fungi are living, their cells are 1N or have only 1 copy of each chromosome in their genome. These cells create a network of cells that we call hyphae, and these hyphae actually have some specialized cells that we call plus and minus, and they will fuse together, and then these particular cells or locations within the hyphae will go through a process that we call meiosis. They will create spores that are released, and these spores can then go through mitosis and create more hyphae cells and networks that make up the fungal organism. And so again, meiosis is very important in these particular organisms' life cycle. The last life cycle we're going to talk very briefly about is in land plants. And land plants have a life cycle that we actually specifically call alternation of generations. And the particular plant that you see here is the fern. And the generations that we alternate between are called sporophyte and gametophyte. The dominant version of the generation within ferns specifically is the sporophyte generation, because those particular cells that make up that organism are all diploid. That's the majority of the type of the organism that we see of ferns, rather than the haploid versions of cells. So on the bottom of the leaves of the fern, the leaves have these things called sporangia, and sporangia go through a process called meiosis again, and they produce spores. These spores are released. They can then go through mitosis and create an organism that looks like this in the picture in the bottom. There's some specialized cells within this little structure, this little organism, that are considered the sperm and the egg. And the sperm and egg will fertilize, create a zygote, and that zygote grows into what we consider the adult version of the fern, or the sporophyte generation. Without meiosis, we would not have that alternation of generations. So very important for the life cycle of the fern. So what we want to do now is we want to go through and look at the characteristics and mechanisms of meiosis. And so just in brief overview of meiosis, there's really 6 key things that I want you to know. Number 1, where does meiosis occur? And this is going to be our germ cells. How many divisions does meiosis have? And it turns out there are 2 rounds of nuclear division. How many cells does it generate in the end? 4. And I'm going to skip number 4. I'm going to come back to that one. It turns out also the 4 cells that are generated, they're genetically unique and haploid, which means they are 1N. And I'm going to come back to number 4 because crossing over is a process that happens during a specific stage of meiosis 1 called prophase 1. And this process, along with a few other mechanisms that happen during meiosis, is one of the major factors or major mechanisms that create genetic diversity in the daughter cells. So we will be going over that very specifically in a few slides. Before we do, I want you to watch a short video on the process of creating these daughter cells in various types of individuals, specifically the male and the female in the human population. So to create an egg in the female, the germ cells will go through a process called oogenesis. And oogenesis works like this.
The ovaries produce secondary oocytes, the female sex cells. During fetal development, stem cells, called oogonia, go through mitosis. Some of these cells develop into 46 chromosome primary oocytes. At the onset of female puberty, tens of thousands of primary oocytes each form part of a primordial follicle in the ovaries. Between menarche and menopause, each primordial follicle develops into a mature follicle, and its primary oocyte completes meiosis 1. Some primary oocytes become secondary oocytes, 23 chromosome cells that begin meiosis 2, then pause. Each month, 1 secondary oocyte is released into the uterine tube. Meiosis 2 is completed when the oocyte is fertilized. The resulting ovum can develop into a zygote.
Now let's look at how spermatogenesis occurs in the male.
Sperm develop in the seminiferous tubules of the testes. Primordial sperm cells, called spermatogonia, lie dormant in the outer compartment of the tubules from the late fetal period until puberty. At puberty, the spermatogonia undergo mitotic divisions, giving rise to genetically identical cells. The cells grow gradually into primary spermatocytes, preparing to divide into cells half their size. Each of these primary spermatocytes undergoes a first meiotic division, forming 2 smaller haploid cells known as secondary spermatocytes. These secondary spermatocytes divide once more, giving rise to 4 haploid cells called spermatids, containing exactly half of the father's genetic material. Through a process known as spermiogenesis, the spermatids change shape, transforming into mature sperm cells. the spermatid's nucleus condenses, the acrosome appears, and the tail begins to grow. Much of the cytoplasm is shed. As spermiogenesis completes, the sperm cells break free from the Sertoli cell lining of the tubules into the lumina. The sperm cells are then stored in the epididymis, where they functionally mature.
Let's do an overview of meiosis. Meiosis occurs in two cell divisions, meiosis 1 and meiosis 2. During interphase, the germ cell will go through G1, S, and G2, and then enter into the stages of meiosis. These stages are both broken up into individual substages, the first being prophase 1, then prometaphase 1, metaphase 1, anaphase 1, and telophase. At the end of telophase, cytokinesis occurs and creates two cells. And then the second phase of meiosis occurs, called meiosis 2. The same names of prophase, prometaphase, metaphase, anaphase, and telophase are used, except this time we use the Roman numeral number 2 to denote that they are different than the first stages of those substages. The ending result is you have 4 different unique, genetically unique daughter cells after this process. Now what I want to do is let's take a look at meiosis 1 in a little bit more detail and figure out what kind of information sets it apart from meiosis 2 and even the process we learned in chapter 6 called mitosis. So a couple of vocabulary terms you're going to need to know are the words synapsis, crossing over, chiasmata, tetrad, and recombinant sister chromatids. So during meiosis 1, during this substage of prophase 1, most of these terms are going to be used because it turns out prophase 1 is an incredibly important process and what creates the majority of genetic diversity in a gamete. What occurs are that all of the chromosomes that are created during the interphase process will come together in homologous pairs of chromosomes, which we call synapsis. When they synapse at a chiasmata, crossing over will occur, and these 4 chromosomes that you see here, we call a tetrad, will move around together within the cell. So the most important thing that you need to know in prophase 1 is that crossing over occurs. And what is crossing over? Well, this is the swapping of genetic information from one sister chromatid to another. When they swap information, we call those recombinant sister chromatids. The other thing is that something really important in prophase or anaphase 1 occurs, and that is after this tetrads line up in the metaphase plate or equator of the cell, anaphase 1 separates the homologous pairs of chromosomes, but still as attached pairs. So there's 2 sister chromatids per pair, and they are pulled apart, or the tetrad is pulled apart. And so each cell that this divides into at this process is considered 2N. So let's look at the second step of meiosis called meiosis 2. Meiosis II has a few things, a few vocabulary terms that you need to know. Interkinesis, gametes, and haploid. So remember, we have our 2 daughter cells from meiosis I. These cells are still 2N, which means they have 2 copies of every chromosome. But if you'll notice in these little arms of these chromosomes, part of them are red, and this one right here is part of it is blue, and that's because these sister recombinant chromatids have swapped information from one another. And this process of meiosis II occurs very similar to what occurs in mitosis. You have the normal prophase functions, prometaphase functions, then you'll go to metaphase 2, where you line up your sister chromatids or your chromosome pairs together in the equator. Anaphase 1, though, or anaphase 2, rather, is where you start to get changes that occur from meiosis 1. And this is, we're going to be separating sister chromatids. This is a very important process. If this process occurs correctly, and then these cells divide during telophase 2 and cytokinesis, then you're going to end up with 4 cells that have a genome that is considered 1N. So you will have only 1 copy of every chromosome. So how does the crossing over process work? Crossing over occurs between 2 pairs of homologous chromosomes. So I call this a pair of homologous chromosomes, meaning that each one of these chromatids are all the same chromosome. So let's pretend like this is chromosome number 1. They have the same type of gene, although one pair of chromosomes, say from a male or its dad, may have a different variation of the same gene than, say, the chromosomes from a mom, but yet the same gene, just variations of that gene. When crossing over occurs, it will occur when the tetrad has been formed. You will swap portions of the arms that are closest together, and we call these recombinant sister chromatids. So that you see here, you've got a recombinant sister chromatid and a recombinant sister chromatid. And the ones that did not cross over are the ones on the outside. The ones on the outside that are called non-recombinant chromosomes. This only happens during prophase 1, does not occur in mitosis, and does not occur in the second stage of meiosis called meiosis 2. Let's watch a short video on this process. I'm going to start this video at approximately 1:50, I think 1:45.
Let's look more closely at each of the division events. Meiosis begins with prophase 1. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis 1. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no 2 siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the two centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are telophase 1 and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis 2. Meiosis 2 is similar to mitosis. The first stage of meiosis 2 is prophase 2. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase 2 and prophase 1 is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase 2, there is no synapsis of homologous chromosomes or crossing over. In metaphase 2, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis 2 began with two cells, and each of those cells were split into two cells, we now have 4 unique haploid cells at the end of meiosis. These cells are gametes. 2 gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Let's now look at comparing mitosis and meiosis. In comparing mitosis and meiosis, meiosis has 2 divisions, while mitosis only has 1. The major differences while the division process is occurring is prophase 1, you have crossing over in meiosis. However, you do not have that process in mitosis. Another important feature that is different about meiosis than mitosis is the separation of the pairs of homologous chromosomes in anaphase 1. Now, the separation of sister chromatids in anaphase 2 versus anaphase of mitosis is very similar, but the outcome of the entire mechanism from meiosis to mitosis is very different. You have 4 cells in meiosis. They're all unique. And in mitosis, you'll end up with 2 cells that are identical. This can be better formulated in a table that is seen in your textbook, where we're looking at comparing both processes. How does DNA synthesis work? Synapsis, the process of actually crossing over, lining up on the metaphase plate, and then the sister chromatids lining up on the metaphase plate. Both of these, the outcome is after the anaphase of that particular major stage. So if we're talking about meiosis in this case, it'll be a anaphase 1 versus anaphase 2. And again, that does not occur the homologous chromosomes, pairs of homologous chromosomes do not tetrad up in mitosis, so that won't occur. And then the overall outcome of 4 cells versus 2 cells that are identical or unique. What happens if there are errors in this entire meiosis process? Well, there are really 2 categories of errors that we're going to talk about in your textbook. And one of them is disorders that are created by changes in chromosome number. And it's by a process called non-disjunction, which we'll go into a little bit more detail. And the second one is by structural rearrangements of each of the chromosomes. And they can happen in multiple ways. And your text really talks about 2 of those out of the multiple ways we can rearrange the chromosomes. So let's talk about the first process, chromosome number or non-disjunction. A normal number of chromosomes is called euploid. So when you have something that has an abnormal number of chromosomes, we call that aneuploid. And we're specifically going to be looking at 2 versions of that, monosomy and trisomy. Monosomy means you've lost 1 chromosome, and trisomy means you've gained 1 chromosome. So what does that look like in the cell? Well, in very briefly, using the separations of divisions of meiosis 1 and 2, you take a 2N, this cell will actually be 4N, sorry about that, 4N, and during meiosis 1 and anaphase 1, you're supposed to have separations of the homologous pairs of chromosomes. Well, if that doesn't happen, and say one side gets all 4 sister chromatids, then that has a detrimental effect in the end of the 4 gametes that you get. It turns out 2 gametes will have 1 extra chromosome and 2 gametes will have 1 less chromosome. If non-disjunction doesn't happen in meiosis 1, but instead in meiosis 2, during anaphase 2, then what happens is one of the pairs of or homologous chromosomes, sister chromatids, do not separate. And so only one of the cells out of the 4 will have an extra chromosome, and one of the cells will have a missing chromosome. However, the 2 that are left over will be normal number of chromosomes, or euploid. We can actually look at individuals and to determine whether they've had a non-disjunction process happen in their chromosome or genome. We do this when babies are born or when we're testing people for abnormalities in chromosome number or even appearance of their chromosomes through a process called karyotyping. And karyotyping allows us to actually take a cell, color each chromosome, and take pictures of them. And what we can do is match them for the homologous pairs and determine if we have the correct number of chromosomes or if we have a missing or extra chromosome. And we can even look to see if they're missing, if we have extra parts within a particular chromosome, something like that. So we have lots of ways to look to see if there are genetic disorders through this process called karyotyping. It turns out that some of the disorders you have probably heard of but didn't realize why or what that disorder was caused from. And most trisomies and monosomies, this means trisomy having 3 sets of chromosomes or monosomies having 1 set of chromosome, are deleterious in some way or negative in some way for that organism. Some human errors in meiosis create syndromes or disorders that I have listed here. Down syndrome is a disorder where you have 3 copies of chromosome number 21. So we call this trisomy 21. Klinefelter syndrome actually is an extra copy of the X chromosome that's a sex chromosome, and this will be a male that has feminine features, and we call that Klinefelter syndrome. We have Turner syndrome, where you actually only have one X and then no other sex chromosomes. We usually put a 0 for that. And then, so you're missing a chromosome here. And then there's Edwards syndrome, where you actually have 3 copies of chromosome 18. These are just a few of examples of aneuploidy or aneuploidy situations. I wanted to point out that one of the ones that most people know the most about is the Down syndrome. And the risk of Down syndrome actually does correlate to the age of the mother. And it turns out about age 37-ish, you start to see a fairly sharp increase in the percentage rates of possible children born with Down syndrome. And so typically people over the age of 37, at least women that are pregnant over the age of 37, will have amniocentesis that can determine the genetic or genome of their organism, their fetus, so that they can determine if that baby will have the disorder of Down syndrome or some other syndrome itself. Now, there are ways that we can look at chromosomes and how they affect development of an organism. And I don't know if you guys have noticed, what's the problem with having an extra X or missing an X when it comes to the sex chromosomes? So obviously normal females have 2 X chromosomes. What's the problem with having a Y? Or if a male has an X and a Y, what's the problem with having another X? And why, if we have males that typically a normal male only has 1 X, why does that create a problem in individuals that don't have a Y but only have 1 X? Well, it turns out that a lot of things that go along with development of an organism during embryogenesis play a role with the sex chromosomes. And so we use dosage-dependent compensations for the presence of the X chromosome in females to combat that overdosage compared to a male. So for instance, remember, females are XX and males are XY. With females having 2 X's, we have twice the amount of dosage of the genes on the X chromosome as the male. So what we're going to do is go through a process called X inactivation. And X inactivation is very readily seen in the example of the cat. And so you have a fairly grumpy cat looking at us here. And it turns out that if you look at the cat, it has several different colors in the coat. And the coat color is actually located on the X chromosome. And so what happens after fertilization, the zygote will divide and you'll end up with an embryo that has several thousand cells. And at some point, all of the cells decide, hey, we're going to take one of those X's and we're going to silence that X and create what we now call the bar body. And that bar body, which we write like this, therefore is silenced. None of the genes on that chromosome can actually be expressed. And only the other chromosome, the X chromosome, genes can be expressed. And that coat color for that other chromosome is what's present in that particular cell. Now, X inactivation is completely random. So in some cells, it may be we're going to turn off the chromosome that has the orangey color within this cell, and so those cells are going to have the color of black. And then some of the cells we're going to turn off the chromosome that has the black color coat, and so we're going to be more orangey or red. And since it's any cell that does this process, all of the descendants of those cells will have the same X inactivated. Now, the interesting thing about that is that process is random after fertilization. And it was an interesting way to show this is through this process of cloning. So it turns out some researchers at Texas A&M University in College Station took the cat Rainbow, who has several different colors in the cat's coat, and cloned this organism using nuclear cloning, and created the kitten that was born here called Cece, or short for copycat. And it turns out if you look at these two, this is supposed to be a genetic clone of Rainbow. Well, they don't look exactly alike yet. If you do a sequencing of their genomes, they have the exact same genome. Well, what happened here? Well, it turns out that X inactivation, because it's random and happens after the organism has created the zygote itself and started going through the mitosis process, growing the embryo, then this is more of a developmental change rather than a genetic change. And so when you clone a cat, you're not going to get the same look coat-wise to that cat because of the process called X inactivation. Let's look at the second kind of disorder or errors that can occur in meiosis, and this is structural rearrangements. And I'm going to talk about two different kinds, one called inversion and the other one reciprocal translocation. So what do these look like? And this mechanism for how this process happens is a little complex, so we're not going to get into that mechanism. We're just going to show the ending result. In inversion, what happens is you take a section of a chromosome and then you flip it and reinsert it back into the chromosome. And so you can tell that the pattern that was here in orange is flipped in this second chromosome that's written here. And this can cause problems because orientation and specific location on an arm of a chromosome can affect the expression of genes that happen to be on that chromosome. So that can cause problems. Another way we can change this structural rearrangement is through reciprocal translocation. And this is where you have two different chromosomes. And what they will do is they will swap the entire arm of their chromosome. So you don't lose or gain genetic information, it's just moved to a different chromosome. Now, it's possible a person that has a reciprocal translocation has no detrimental effects to their organism, their individual bodies. However, that can cause detrimental effects to their offspring. One particular example is a particular disorder where you have a chromosomal rearrangement that's the reciprocal translocation of chromosome number 5 with another chromosome, say like chromosome number 11. In individuals that have offspring that get chromosome number 5 that has had a portion of it removed and swapped with number 11, it could potentially lose portions of chromosome number 5. Another way is a deletion of portions of chromosome number 5, which was not one of the types of rearrangements we had originally discussed, but it is a possibility. This particular disorder is called Cri-du-Chat. And Cri-du-Chat is a disorder that when the baby is born, there's very distinct physical features that you can use to help you determine whether or not someone has this disorder. One thing is that the larynx of the baby was not fully developed, And it turns out when they cry, they sound like a cat's meow, which cri de chat is French for the cry of a cat. They have a very small round face. Their cheeks are pretty plump and full. They have hypertelorism, which is their eyes are set a little bit further apart than the norm average. They have a very prominent supraorbital arch, which is right above their eye. Their ears tend to be set a little low. Their teeth might be misaligned. They might have cardiac disorders. They have developmental delays. They're not able to walk as quickly as normal babies. There's lots and lots of different things that are components of this particular chromosomal rearrangement called cri de chat. If you want to know a lot more information about it, it's actually a fairly interesting disorder to study up on. I linked a YouTube video that you could watch that's about 10 or 15 minutes long about this particular disorder, and that could give you a little bit more information if you're interested. And so that brings us really to the end of chapter 7, where we've looked at a lot of how meiosis works and how that can then affect an organism if it's done incorrectly. So what we should be able to do now is be able to explain variation among offspring and how that makes them advantageous in their population, be able to describe your 3 types of life cycles, be able to describe how cells and chromosomes behave in mitosis and that mechanism of mitosis itself. We can compare mitosis and meiosis, which is very important, and how errors cause disorders. Errors in meiosis cause disorders. So your next step is, I would like for you to read the chapter, complete your chapter learning guides. Then what I would like for you to do is try to make your own notes from the PowerPoint, which is posted, your chapter readings and the learning guide together. Then create some materials to help you learn the vocabulary and concepts that are brought into this chapter. Flashcards and diagrams are a really good way to do that. You look at some more YouTube videos on meiosis, which I have posted in Blackboard, and maybe get to with a study partner to learn and teach each other together. That brings us to the end of chapter 7.